Thank you so much for your patience this afternoon. It's very much appreciated while we work through some technical issues. Today, I want to welcome you to today's webinar, Understanding Fecal Coliforms and E. coli Testing Methods for Wastewater in Massachusetts. I'm your moderator, Nancy Leonard from IDEXX Water Testing. There will be a question and answer session at the end of the presentation, but feel free to submit your questions at any time during the webinar. You can use the Q&A tool at the bottom of your screen and type in your questions into the chat box. We'll try to answer as many of your questions as possible, but if we run out of time and you still have questions, or if you're watching this as an on-demand viewer, you may submit your questions to water@idexx.com. This webinar is being recorded, and a link with the recording will be emailed to everyone who registered along with a copy of the slides. This webinar will introduce you to indicator bacteria, review the enzymatic test methods for wastewater, quality control, QA and QC, and discuss specific wastewater regulations for the state of Massachusetts. I'm happy today to introduce our speakers. Our North American Regulatory Affairs Associate, Jody Freimeyer, will be walking you through the specific wastewater regulations for the state of Massachusetts. Jody received her undergraduate degree in preclinical naturopathic medicine from Johnson State College in Vermont and is currently studying at the University of Nevada to earn her master's degree in public health. She's been a part of our water business for 5 years and works with a variety of regulatory agencies to help obtain regulatory approvals for IDEXX methods. She also participates in several standards organizations, including the NELAC Institute, the American Standards, and ASTM. Before we get to the regulations that affect your state, please join me in welcoming Sean Dubois. Sean has a BA in biology and a BS in environmental science with a concentration in marine biology and marine ecology. Sean has been a part of our water business for 17 years, working in research and development, and in his current role, managing the product support group. Welcome, Sean.
Thank you, Nancy. So today I'm going to be going a bit over wastewater testing, and going over part of it, going to go over a little bit over what is indicator bacteria, why we look for them, how we look for them, different methods, which include, of course, the IDEXX methods. So a little bit about indicator bacteria. So part of the problem of looking for waters that are contaminated is that the number of pathogens that could be possibly in contaminated water are numerous. They can be very difficult to actually detect, and being difficult to detect and being pathogens themselves, they can also be kind of dangerous. So the best thing to do is look for an indicator of contamination, and these indicators will have certain characteristics. So what do we look for requirements? Well, they should be present when the pathogens are present. So if there is a contamination event, then these organisms, particularly these bacteria, should also be present at the same time. Conversely, when there is no contamination, they should not be found. They should only be occurring when there is a contamination event and not naturally occurring in the environment. They should be present in high numbers in the contamination event. So when it comes to viruses and other pathogens, they can tend to be in concentrations that are lower and makes it a little more difficult to find. So a good indicator would be an organism that would be found in high numbers at the same time that these pathogens would be found. They should also be able to survive longer in the water. So basically we want to be able to tell if a contamination event has occurred so much time after it has happened. So that way we're not limited to time-wise. They should be easy and safe to analyze. Once again, pathogens we should want to handle as little as possible. So a good indicator would be something that we can easily take care of and use in the labs. We also want rapid results. So we want to know as soon as possible that there has been an issue, so that way we can begin fixing it and treating it. Of course, inexpensive is always good, and most importantly, accurate. We want to be able to know what we're detecting and know that the results that we're getting are correct. So when we think of a good indicator organism, the one that will tend to pop up first will be E. coli. E. coli is basically a major inhabitant of the intestines of most warm-blooded animals, including birds and people. They are shed in high numbers during fecal contamination, and they are only present when there has been a contamination. They're not found naturally occurring in the environment. So this makes it a true indication of a fecal contamination event. A little bit more about them. They are gram-negative bacteria and thermotolerance. They're able to survive in elevated temperatures. So it kind of checks off all those boxes of what makes a good indicator organism. Once again, not naturally found in the environment, found during contamination events. You got to find them in high numbers, and they're relatively long-lived once they're in the environment. Focusing a bit more on the general types of bacteria that can be tested for contamination, what kind of started off is the Coliform group. And this group is bacteria that tend to be found in fecal material, but can also be found in the environment. And this grouping contains several different genus, including Klebsiella, Citrobacter, Enterobacter, Escherichia for E. coli. There's a subgroup of Coliforms that are known as Fecal Coliform, or that's what they were called. These were bacteria that were deemed to be the best indication of a fecal contamination. Basically, the way this group is kind of decided by is selected by temperature. So in this case, for this kind of testing, you're looking at 44.5 degrees Celsius, plus or minus 0.2. So it's a very high temperature, a very tight tolerance. And it was believed that this would be the true indication of a fecal coliform. However, the truth is there are some environmental bacteria, such as Klebsiellas and some Citrobacters and Enterobacters, that are actually able to survive at these elevated temperatures. But these bacteria might not be actually coming from a fecal source. They could just be naturally occurring in the environment. So they're not truly a fecal coliform. And likewise, not all E. coli are thermotolerant, so they're not able to survive at these elevated temperatures. Most can, but some can't. And other ones might have a variability of tolerance. So you're truly knocking out a fecal indicator of E. coli by this temperature. So it's not truly fecal coliforms. The better term is thermotolerant. But from here on out, we'll just use the term fecal coliform because that's what most of us are used to hearing. So when it comes to testing wastewater, there has been a kind of evolution over time of the methods in order to detect a contamination event in the wastewater. So we kind of start off with multiple tube fermentation. So this process, it tends to be lengthy. To get a final result, a final confirmed result, we're looking up to around 96 hours. So we're looking at several days before we can finally get a confirmed positive. There's a lot of steps. We're looking up to 60 steps in order to run this method. It does require some amount of knowledge and a lot of patience and a lot of material. So it's good that there's a method able to be utilized for detecting in wastewater. However, it's not exactly meets all that kind of criteria of being easy and cost-effective. So next came membrane filtration. So the number of steps have been reduced from around 60 to up to 20. So getting a little faster, and results are coming out about 24 to 48 hours after testing it started. So it is faster, but still not quite there, and a lot of confirmations tend to be required for the different methods. So an improvement, but still not the best. And finally, we come to enzymatic methods, which includes Coliert-18, and Coliert-18. So now we're looking at a confirmed result within 18 to 24 hours, depending on the test, with up to only 8 steps for quantification. So a further reduction in the amount of steps in order to run those samples, and confirmed results within a day. So much of vast improvement. So from here, going into the different IDEXX products, we have Coliert, Coliert-18, and Coliert. The way that the IDEXX products work is on the same general principle that you have a reagent that contains basically like 3 groups of materials. You have your food, to help the target bacteria grow. There are suppressants to prevent other bacteria from growing. And then finally, the nutrient indicator. And this nutrient indicator is what will be utilized by the target bacteria that will say whether or not that organism is present in that sample. So starting off looking at Colilert and Colilert-18, there are actually two indicators in these products, one for the detection of total coliforms and the second one for E.coli. But first, for total coliforms, we have O-nitrophenyl beta-D galactopyranoside, which is specifically going to be utilized by the enzyme beta-galactosidase. So total coliforms have this enzyme and therefore are able to utilize this nutrient indicator. And the way this works is that there is the food, the beta-D galactopyranoside, is bound with an O-nitrophenyl group, and this is actually the indicator. While these are bound together, the appearance is colorless. However, once the enzyme is used and that food group is basically eaten, the indicator is cleaved off and is released, and it will, through hydrolysis, it will turn into O-nitrophenol, which then turns yellow. So you go from clear to yellow in the presence of total coliforms and fecal coliforms when it comes to Coliform-18. The second indicator is 4-methyl unbifferyl beta-D glucuronide, or MUG, and this indicator looks for an enzyme that's specific to E. coli. So E. coli, once again, is a total coliform, so it will utilize the O and PG. At the same time, it has this beta-glucuronidase enzyme that's able to utilize the MUG. So in this case, once again, it's the same principle. You have a food, and attached to that food, with that food is a indicator, in this case, a 4-MU. When these are bound, the media will not fluoresce when exposed to a UV light. But once that enzyme utilizes this nutrient substrate, the enzyme is released, hydrolyzes, becomes 4-methyl unbifferone, and then when exposed to a UV light, will fluoresce and give off a glow. So total coliforms will have a yellow appearance in the media, whereas E. coli will have yellow with fluorescence. Our third product, Enterolert, utilizes the same principle, except this time it's a 4-MU beta-D glucoside is the nutrient indicator, and enterococci contain the enzyme glucosidase, which once again, once utilized, will go from non-fluorescence to fluorescence when exposed to a UV light. So we have the reagent, which will basically is the food and the indicator for the bacteria in the samples. So we can say yes or no, it's there. Next part is how do we quantify these samples to understand how much bacteria is actually present in those samples. So for that, basically we require the Quantitray system, which includes the Quantitrays. We have two types, Quantitray and Quantitray 2000. I'll go a little bit more into those later. The Quantitray inserts, which are used with the Quantitray sealer in order to seal the trays and actually create the quantification device. So the process to run samples is pretty simple. Basically, you'll take your sample. To that, you will add the appropriate reagent. So in this case, we'll talk about Colilert or Colilert-18. You will add the reagent to the vessel, shake, allow it to dissolve. Once it's dissolved, it gets poured into the Quantitray, which at this point is basically acting like a bag. Be poured into the tray. That gets added to the Quantitray insert, which then goes through the Quantitray sealer, and through heat and pressure, it activates an adhesive that's on the back of the tray that will then seal the back of the tray to the plastic front, and you'll have a sample that's distributed in the Quantitray. So when it comes to Colilert and Colilert-18, each of these products are incubated at 35 degrees for the detection of total coliforms in E. coli. So 35 plus or minus half a degree Celsius. Colilert is a 24-hour test with results valid up to 28 hours. Colilert-18 is an 18-hour test valid up to 22 hours. So they're both incubated at the same temperature. They're going to give you the same reaction. You're looking for the same signal output. The only difference here is the time to detection. So 24 hours for Colilert, 18 hours for Colilert-18. So the tray gets placed into the incubator after you take it out for the appropriate read. What you're going to do is look for that reaction of those indicators. So you're looking for yellow, which indicates a positive for total coliforms. If you find any wells that are positive with yellow, the next part would then be to take that tray and place it under a UV light and look for fluorescence. So if you find fluorescence with yellow, that indicates a positive for E. coli. Once again, this is when you're running Colilert and Colilert-18 at 35 degrees, looking for total coliforms in E. coli. So yellow, positive for total coliforms, yellow with fluorescence, a positive for E. coli. Colilert-18 can also be run as a fecal coliform test. So in order to do that, the process is still the same. You would take your sample, add Colilert-18, shake to dissolve, add to the tray, seal. And then these trays, if you're running for fecal coliform, would be incubated at 44.5 degrees plus or minus 0.2 degrees Celsius. And this is really important. Fecal coliform testing requires a very tight tolerance in order to get the appropriate result. If the temperature is below this range, so below 44.3, then the sample will become too cool and it will allow non-thermotolerant bacteria to grow, which could inflate your numbers. And if you're above 44.7, the temperature will be too high, which could reduce the counts by knocking out some of those thermotolerant bacteria because it will be even too high for them. So it's really important to maintain the 44.5 plus or minus 0.2 degrees when testing for fecal coliform. And you can see in the image here, we also suggest using a water bath for fecal coliform testing. Water baths are able to maintain that tight temperature tolerance a lot more accurately than most air incubators can. So using a water bath will help ensure that that temperature is being maintained. And what you want to do with a quantity tray with Colilert-18 for fecal coliform testing is take that tray and submerge it completely below the surface of the water in the water bath. And you want to weigh it down to make sure that during the 18 hours of incubation it remains weighed down. And you can see in the picture here there's some weighted rings, and these would be the more appropriate kind of items used to weigh down the trays. What you don't want to use is something that would be like a rock or brick, something that, well, it will weigh it down, has a good chance of also breaking it. But also you want to not use something like a beaker full of water or a bottle. That kind of stuff can throw off the thermodynamics of the whole system, which might create some issues with results. So you want to make sure you use the appropriate way to weigh down the trays and keep them below the surface of the water in the water bath. So once again, this is Colilert-18. So you're looking at an 18 to 22 hours for your final results. And once again, since we are looking for fecal coliform, we're only looking for yellow color. So yellow color indicates a positive for fecal coliforms. Because as mentioned before, not all fecal coliforms are going to be E. coli, and so therefore you just want to look for that yellow color. When reading Colilert and Colilert-18, a comparator can be used. A comparator will provide the minimal amount of yellow and fluorescent signal to call something a positive. So any yellow color or fluorescence that's equal to or greater than the comparator is considered a positive result. So moving on to Enterolert. Basically, Enterolert is run exactly the same as Colilert and Colilert-18 in that you'll just take your sample, add the reagent, dissolve, add to your quantity tray, and then seal. For Enterolert, there's a different incubation temperature. It's 41 degrees plus or minus half a degree. And then the time for results are 24 hours with results valid up to 28 hours. And then from there, after the 24 hours, you remove the tray from the incubator, exposed to 365 nanometer UV light, and looking for blue fluorescence. Blue fluorescence indicates a positive for enterococci. So at this point, you've gone through, taken all your trays out, read the results, count the number of positive wells, depending on which reagent you're using. The next part is to figure out your most probable number, your actual result from that sample. So number of positive wells are counted, and then you refer to the MPN table that's included with the tray. So this is where the difference between the two trays comes in. So the 51 well quantity tray will give you a counting range of up to 200 MPN per sample. The Quantification Tray 2000 will give you counts of over 2,000 for one sample. And this is done because, as you see in the picture here, there's actually two different size wells, a large set of wells on the top and then small ones on the bottom. So the 51 well will have 51 wells, 50 of them being of a large size with one overflow well on the top. All those are counted as your 51. The Quantification Tray 2000 has the 49 large wells on the top, which includes that large overflow well on the very top, and then 48 small wells on the bottom. So you count the number of positive wells and refer to the MPN table. So this shows an example of the 51 quantity tray, well quantity tray MPN table. So you can see here, if you had 4 positive wells, your result would be an MPN of 4.2 per 100 mLs. With the Quantification Tray 2000, you count out the number of large positive wells and the number of small positive wells to obtain your result. So in this case, if you had 48 large and 25 small positive wells, that would result in an MPN of 344.1 for that sample. So speaking of MPN, one of the things that we often have gotten questions of over the years, at least definitely since the 17 years I've been here, is the MPN versus CFU. For people who are used to counting plates and reporting out the results in CFU coliform units, the question is, well, I'm now going to report on MPN. Is that an issue? Because are they different? And the simple answer is they're not different. What that CFU and MPN refer to is the method that you're using for testing. MPN, most probable number, is used for when reporting results that are done in a liquid media, such as Colilert, whereas coliform units, or CFU, are when you're doing on a solid media, augers. So it's more of a label that's associated with the result as opposed to giving a different, meaning something different. So reporting out as MPN versus 100 ml or CFU per 100 ml basically will give you the same result, just giving the differentiation of what method was used in order to obtain it. And what's important to know is that both MPN methods and plate methods for CFU, they both have a 95% confidence range associated with them, because there's always some error that can be done when you look at the result. You got Poisson distribution, it's pretty evenly distributed. So both of them have that. Nothing is an exact answer. Quantity tray, one of the many advantages over quantity tray over memory filtration is that, for the most part, dilutions aren't going to be required, because the counting range of the Quantification Tray 2000 will give you counts of up to over 2,000. So for the most part, dilutions might not be required, whereas with plate methods, you'll definitely have to do dilutions. So the question becomes, if you're going to have a result that you know will be outside of using the quantity tray, can you do dilutions with the test? And the answer is yes. Basically, what you want to do is perform any dilutions prior to adding the reagent to the sample. So create whatever dilution you require. Then from there to the diluted sample, which is then brought to 100 ml total, you would add the reagent and go through the same steps, and then incubate results, take them out, read them, and basically correct for the dilution that's done for the result. So, for example, if you did a 1 to 10 dilution, 10 ml of sample, 9 ml of diluent, and you got an answer of an MPN of 344, and that means your final result per 100 ml would be 3,440. So basically, you have to remember to do the dilution correction factor. All right, now I'm going to go a little bit into the QC when we talk about QC for our products and other ones. One thing that we'd like to point out is that there's a lot less QC that's required for the enzymatic methods compared to plate methods. The best example is when we compare Coliert-18 for fecal coliform testing, comparing that to MSC. MSC has a lot of QC steps that are required now when properly running the protocol. So basically, with both methods, Coliert-18 and MSC, there's should be positive negative controls done with the addition of any new lots. But after that, for Coliert-18, there's no other QC involved. But when we look at MSC, of course, there's now verifications that need to be performed on each lot, up to 10. You're running anywhere from as little as 2 blanks per sample run, sometimes more depending on how many samples you're running. And then on a monthly basis, there now has to be 10 colonies verified in each one. So that's in addition to any other QC for when new batches of plates are either made or received in that need to be done. So there's a lot more QC now that's expected for plate methods as opposed to Coliert-18 or any of our other products where you're looking at just doing one QC event for each new lot of material that is purchased. On that, IDEXX does provide several QC that can be done on our products, but we also note in our inserts that you can run positive negative control using bacteria obtained from culture collections such as ATCC. But once again, IDEXX does provide IDEXX QC products, and there are separate kits for each type of each test. So here we have ones for our TCEC, so Coliforms and E.coli, and you'd get 3 different bacteria. You get a E.coli positive, you get a Coliform positive with Klebsiella varicola, and then a non-target with Pseudomonas aeruginosa. For Fecal Coliform, once again, you get E.coli and Pseudomonas, and then for enterococci kit, you get E. fecalis as your positive control, and then a gram-negative and a gram-positive negatives with E.coli and Staphylococcus. Speaking more on quality, what's really important is that for your records, for any kind of audit or just to have the certificate of analysis that comes with each of our products. So we, all of our products go through different QC quality controls testing, and all of that testing gets recorded and is placed on our certificate of analysis. And all those can be obtained on our website. There's a link here in the presentation that will take you right to the page. And basically, it'll be a box to fill in the lot number of the product. Once you enter that, it will bring up a list of all the products that are associated with that lot number. And then clicking on any of those will then provide a PDF of that certificate analysis that can then be printed off or saved for your records. One of the things that we definitely pride ourselves on is the quality of our products. So a lot of work goes into ensuring that everything that we release here is of a high standard. So whenever there is a new batch of reagents created here in Westbrook, that is, there are samples collected throughout the entire run of that lot. Those materials then go to our quality control team. They will run a battery test against that lot. All that information is collected. Once everything has been confirmed to be working appropriately and is passed, that is all recorded, and then that goes to our quality assurance team. And they will review all of our records to make sure that everything has been done correctly and everything's filled in. And then once the QA group has approved it, that lot is then released for distribution. So there are multiple steps that we have during our processes to make sure that everything that we're releasing is of a high quality. To further say that, IDEXX as a whole does have several ISO certifications, including 9001 and 14001. And then for our water QC lab, we actually have ISO cert for 17025 for the majority of our products. And basically it just goes to show the extra effort that we put in through our QC team. And this actually does help with some countries that follow the ISO standards. It actually can reduce some of their QC that's required because of our certification. So once again, all of our products, whether the reagents, the quantitrays, vessels, IDEXX, all of that has an associated certificate of analysis that can be obtained on our website for all products. And once again, it will tell you all the testing that's done and whether or not it passes, and go from there. But that's it for my end.
Thank you so much, Sean. Really appreciate the information. And just as a reminder, if you do have questions for Sean, if you would like to type those into the Q&A chat box at the bottom right-hand side of your screen, please do that before you forget your question for Sean as we move on to Jody. So welcome, Jody.
Thank you, Nancy. Hello, everyone. So the U.S. EPA drives regulations in Massachusetts, just like it does for all other states. Specifically, the EPA's Clean Water Act was created to control the discharge of water pollutants to surface waters, and these wastewater regulations can be found in the Code of Federal Regulations at 40 CFR Part 136. Now, every so often, these regulations are updated through a process called the Method Update Rule. And the Method Update Rule done by the EPA in August of 2017 included the EPA's final approval for IDEXX's Colilert-18 Fecal Coliform method for wastewater effluent testing. After the EPA had its method update in 2017, states that have primacy can either incorporate the federal rules into the state rules, or the state can create rules that are equivalent to the federal rule. But if the state does create its own rule, it must be as stringent as the federal rule. It can't be less stringent. And just to quickly mention, there are a few hyperlinks within some of the regulatory slides. So once the presentation is available, if you'd like to review any of the regulations or contact links, you can click on the hyperlinked word that's outlined in blue. And here on this slide, the hyperlink will bring you to view the EPA's Code of Federal Regulations 40 CFR Part 136. So wastewater effluent monitoring is enforced through the National Pollutant Discharge Elimination System Permit Program, which is referenced as NPDES for short. In Massachusetts, currently the U.S. EPA Region 1 is the permitting authority, but NPDES permits are usually issued by both Region 1 and the Massachusetts Department of Environmental Protection. NPDES permits regulate wastewater discharges by eliminating the amount of pollutants that are allowed to be released, which these limits are based on and must be compliant with the Massachusetts Surface Water Quality Standards and federal regulations. So for testing requirements, NPDES permits usually state a general allowance for the use of any analytical test method included at 40 CFR Part 136. However, it is possible that some wastewater permits could list a specific test method instead of stating the general allowance. So if there were questions on an NPDES permit, for example, how an individual permit is written, or if a test method is listed, it's best to contact the permit writer included on the permit, or you could also reach out to the U.S. EPA New England contact provided here. So since the EPA has adopted the method update rule, this change affects wastewater laboratories in Massachusetts by allowing more analytical test methods that could be used for NPDES permit fecal coliform monitoring requirements. This revision can be really helpful to laboratories since, as previously mentioned by Sean, Colilert-18 reduces quality control steps, reduces hands-on time, makes demonstration of capability easier for both a new analyst or for any ongoing DOCs that might need to be continuously done over time, and it allows for more reliable data. So if a laboratory is interested in changing an analytical test method, we're going to be going over at a high level how this can be done. Laboratory accreditation for wastewater testing is not mandated in the state of Massachusetts, meaning some facilities or laboratories may or may not be certified. For laboratories or for facilities that are not certified, it's still good practice to conduct some sort of method verification when adopting a new test method. So some of these steps might include conducting a parallel study between a new and existing method, possibly doing a 7 to 10 split sample comparison, writing a standard operating procedure for the new method, And as part of the SOP, you would want to include the quality control requirements, which those are listed within the Coliform-18 package insert. If you did feel like you would like some help creating an SOP, here at IDEXX we do have an example SOP document that is available by request. Other steps you might want to consider when changing an analytical test method might include, if you have a laboratory quality assurance manual, it might be good to just review that and make any necessary updates. And you would also want to consider reviewing and verifying if there are any DMR QA study requirements that might need to be completed. Now, for accredited laboratories, you would want to follow the Massachusetts Department of Environmental Protection Laboratory Certification Program for requirements for monitoring your current scope of accreditation, which these steps would include submitting an application packet that contains a quality assurance plan, a standard operating procedure, a completed modification of certification for microbiology laboratory LES-04-EA form, and a completed transmittal form. Also, you'll want to include the results of 2 successful proficiency tests. Additionally, the laboratory will need to complete a payment packet, which is to include a copy of the completed transmittal form, just a copy of the one that was completed in the application packet, and a payment for the laboratory certification permit application and compliance fees. Which, just to also know, we have put together a document that outlines today's regulatory content in the form of a method adoption guide that is specific to the state of Massachusetts. This informational guide also includes the example standard operating procedure document that I mentioned on the previous slide. And if anyone would like a copy of the informational guide, please reach out to us and just request one. So today I talked a bit about Massachusetts regulations, and just to wrap up today's regulatory portion, I'd like to share part of what I do, which is I follow various state rules and regulations to see how they periodically change over time. I'd like to provide just a little bit of information on how easy it is for anyone to follow and even participate in Massachusetts rule changes. Since anyone from the public can comment on a proposed rule change, and comments can be quite impactful to rulemakers, especially when they come from stakeholders such as wastewater utilities or laboratories, since you're the ones who do this type of work and you might be directly affected by any rule changes. A comment submitted to the state can include things like your opinion on the rule subject, any data you might like to share that might be impactful pertaining to the regulation that you're commenting on, or how the proposed rule change might impact your daily work. Typically, a public comment phase is open for about 30 to 60 days, but you can sign up to receive email notifications on various rulemaking activities so you can watch and track what's happening in your state. On this slide, if you'd like, you can click on the hyperlink and it'll bring you to a place where you can sign up for your state. And this screenshot just kind of shows how easy it is to sign up. You can just type in your email address and you'll be all set to go. And that concludes what I have for the regulatory portion today. Thank you.
Great. Thank you so much, Jody. Appreciate the information. We have gotten a few questions in on the Q&A section. Just as a reminder, if you do have questions, please type them in at the bottom of your screen, and we'll get to as many as possible. So first question looks like goes to you, Jody. Does a commercial lab need to add Colilert-18 fecal coliforms to their accreditation scope?
Yes. If the laboratory has laboratory accreditation, then yes, they would need to add it to their scope. Yes.
Okay, and the next question is for you, Sean, is if I have a sample that has turbidity or sediment, can I use that sample?
Yep. Samples with turbidity and sediments could be done. We suggest that if there's any question about the turbidity interfering with the read, then it's probably best to do some kind of dilution, and that would take care of that turbidity and be able to make it a little clearer to read if there's any kind of concern that they would have any kind of color. And sediment, same thing. Basically, you don't want to have too much sediment in there, especially if you have like large, hard material. That could cause an issue with the tray. But for the most part, it should be fine.
All right, great. Thank you, Sean. And I've got a couple of questions that have come in about the method adoption guide. Jody, I know that you mentioned it in the presentation. Can you just remind us again how we can get a copy of that?
Yep. You can submit inquiry in the chat box or Q&A box on today's webinar, or you can send us an email at water@idexx.com, and we can certainly send you one.
Okay, great. Thank you. And Jody, another one for you. What do I do if my permit states a specific method?
Yeah, that's a good question. So if MPDES permit lists a specific test method, the first thing you would want to do is contact your permit writer listed on the permit, and then you can ask what needs to be done to have the method changed.
All right, great. Thank you. Sean, question for you. Are there environmental bacteria that can show up as positive on Colilert even though they aren't fecal coliforms?
So if referring to fecal coliform testing at 44.5, and that would be Colilert-18, just to make sure we clarify that. But when it comes to the bacteria that we're going to detect, so Colilert and Colilert-18 are designed to detect all total coliforms in E.coli, whether or not they're from an environmental source or if they are from fecal. So if that environmental bacteria that you're referring to is a coliform, then yes, it will detect that. And then, so yeah, it will detect that. If it's not, if it's an environmental bacteria other than a coliform, then it should be suppressed by the system and would be detected.
Okay, and just as a follow-up for that, how common is that?
Well, when it comes to coliform testing, it should be detecting coliforms all the time.
All right.
It's an environmental strain, otherwise it should be not seen.
All right, thank you. Sean, we talked about a comparator for Colilert, and is there a comparator for, there's two comparative questions for you, sorry. Is there a separate comparator for fecal coliform interpretation?
Nope, there is just the one comparator, and that comparator is for both Colilert and Colilert-18. Provides the yellow and fluorescence, and then if you're doing fecal coliform testing, then you just use the yellow.
Okay, and another comparative question for you, is there a comparator for Enterolert?
There is no comparator for Enterolert. When it comes to Enterolert, if there's any kind of question whether or not something is positive, we would suggest to run a negative control. And if you see any fluorescence compared to a negative control, then that would be considered a positive if there's any question.
Okay. Another one for you, Sean. Why are there smaller wells at the bottom of the Quanti tray?
So for the Quantification 2000, the smaller wells basically increase the counting range. It's like a built-in dilution in the tray. So having the two different size wells increases the counting range, allowing us to have over 2,000 for a 100 ml sample.
Okay. Another one for you, Sean. Is it okay to use an air incubator instead of a water bath?
If we're talking about fecal coliform testing, then we would highly suggest just focusing on the water bath. Since that tolerance of that 44.5 plus or minus 0.2 is really important, unless you have a really good high-end air incubator, sometimes it's very difficult to maintain that plus or minus 0.2. So a water bath is best for fecal coliform testing.
All right, great. And it looks like I only have about one more question, maybe two, if I can combine these two. Is Colilert okay to use for fecal coliforms and not Colilert-18?
The only approvals we have are for Colilert-18 for fecal coliform testing. So just Colilert-18.
So, I'm sorry, you said no approvals for Colilert,
yep,
only
just
Colilert-18. Okay. All right, and then, let's see, I'm just checking questions to make sure we got them all. Sean, if a yellow well fluoresces, can I report it as an E. coli positive?
Can you say that again, sorry?
If a yellow well fluoresces, can I report it as an E. coli positive?
Yes, if you're running the test at 35 degrees, then a yellow well that also fluoresces is considered an E. coli, correct?
Okay, and looks like one more for you, Sean. is it correct that Colilert provides both total coliform and E. coli without any difference in cost?
Correct, because there's no difference because it's all contained in one test. The only difference is that when you're running it, you're just looking for yellow. That'll give you the total coliforms, and then look under UV light. So the only additional cost is that you would have to have a UV light in order to be able to see the fluorescence. So it's the same test, it's all in one, so you're just buying one test and you get two results out of one test.
All right, great, thank you. And it looks like we got some coming in through my private chat, but I think we got all of those. So thank you so much, Jody and Sean. We really appreciate your time. And for those of you on the phone, we appreciate your time as well. If you have any questions following the webinar, please do send them to water@idexx.com, or if you have any feedback for us for these webinars or any future webinars that you may like to see from IDEXX, again, the email address is water@idexx.com. Thank you so much for joining us today, and enjoy the rest of your day.